Today, we are diving deep into a field that honestly sounds like something out of sci-fi, but it's very real. We're talking about engineering DNA itself into two-dimensional materials. So our mission today is to figure out how our own genetic blueprint, DNA, is being, well, engineered incredibly at the nanoscale to create these revolutionary, flat structures. You'll find out about its surprising properties, what it can actually do, and how it compares to other advanced materials you might have heard of. It's really a new frontier.
Absolutely. And for this one, we've really dug into a pretty substantial stack of sources. It's a mix of, groundbreaking research papers, some really comprehensive reviews, foundational stuff too. Gives us a solid look at this really fascinating, fast-moving area.
All right, let's jump right in then. So most people listening might be familiar with 2D materials like, say, graphene, these super thin sheets, right? Often just one atom thick.
Yeah, thanks.
Known for their, unique electronic, mechanical, optical properties. We hear about them a lot. But applying that same idea to DNA, what's the sort of basic idea? How can we treat something biological like DNA as a building block for 2D materials?
Right. Well, that's where DNA and nanotechnology really comes into play. The core concept is actually brilliantly simple in a way. DNA just has this natural ability to self-assemble. It wants to put itself together.
Okay.
And it does this because of its incredibly precise base pairing rules. A always links with T, G always links with C.
Yep.
Scientists can basically use this like programming.
Like molecular code.
Exactly like molecular code. It lets us direct individual DNA strands to connect in very specific ways. We can build structures so tiny and intricate. It kind of redefines nanoscale engineering.
So, okay, if DNA is like this programmable molecular Lego, what's maybe the most powerful technique people are using to build these 2D shapes? I'm thinking DNA origami, is that the main one?
Yeah, DNA origami is definitely central to a lot of this. Imagine you take a really long single strand of DNA. Often it comes from a virus, actually, just because it's a convenient long piece.
Right.
And you use that as a sort of scaffold. Now, instead of just letting it float around, you add hundreds of much shorter staple strands.
Staples.
And these staples are designed really precisely to bind only to specific complementary spots along that long scaffold strand.
So they grab onto it.
They grab on, and as they bind, they act like tiny molecular clamps. They pull and fold that long scaffold strand into whatever shape you predetermined. It can be a really complex 2D shape.
Wow. It's like molecular paper folding, isn't it? The amazing thing there seems to be the control you get. You're not just hoping things stick together.
No, not at all.
You're literally dictating the exact structure, almost atom by atom. That seems incredibly hard with other materials.
Precisely. That programmability is key. And, the cleverness goes even further. There are various ways to actually fabricate these things beyond just that general origami idea. We have methods like tile-based assembly. This uses repeating units, often called double crossover or DX tiles, sometimes triple crossover, TX tiles.
Okay, so like building with identical bricks.
Kind of, yeah. They're great for creating large repetitive patterns like lattices. Then you still have scaffolded DNA origami for more unique, one-off, intricate designs. There's even algorithmic self-assembly for making structures that can, compute things, and DNA bricks, which are even smaller, more modular units.
So many options.
Yeah, and the beauty is, again, that precise sequence programming lets you achieve almost any 2D shape you want, and even complex 3D shapes, too. And just to keep them stable, because they can be delicate.
Right, I was wondering about that.
Yeah, we often use things like magnesium ions, other colloanes or polyamines in the solution. They help shield the DNA charges and keep the structure together.
That level of control is it's genuinely mind-bending.
Okay, let's talk properties. What makes these DNA materials special? You mentioned programmability. What does that really let us do?
Well, it's like having the ultimate customization tool, but at the nanoscale. That programmability gives you an almost unprecedented degree of control over the size, the exact shape, and even what's on the surface functionality.
Just going to add things to it.
Exactly.
Yeah.
You can decide precisely where every component goes. You can attach specific molecules, maybe tiny sensors or drug molecules, at exact locations on the DNA structure.
Yeah.
It's a level of precision other materials really struggle with.
Okay, so we can design the blueprint perfectly, but are they strong?
Yeah.
What about their mechanical properties? Are they rigid, like a tiny sheet of metal, or floppy?
It's interesting. Generally, DNA origami structures are quite rigid. That's because the DNA helices are packed together so densely.
But, and this is cool, their stiffness and flexibility can actually be tuned. Researchers can deliberately put in regions of single-stranded DNA, which are much more flexible.
Ah, like flexible hinges.
Sort of, yeah. To introduce controlled bending or movement points. So things like bending rigidity, how stiff it is, depend directly on the type of lattice you build and how densely packed the connections, the crossovers are. It's a real balance between rigidity and design flexibility.
That's fascinating. Precise, tunable. And you mentioned movement. Can these DNA structures actually move or change shape on command?
Oh, absolutely. That's actually one of their most powerful aspects. DNA nanotechnology lets us create structures that are truly dynamic and reconfigurable. We can even build autonomous molecular machines.
Machines made of DNA.
Yes.
Yeah.
And the crucial mechanism behind this dynamic behavior is something called DNA strand displacement reactions.
Okay, what's that?
Think of it like a molecular game of musical chairs. One DNA strand can come in and competitively kick out or displace another strand that's already part of the structure. When that happens, the structure can fundamentally change its shape, or maybe expose a new active site, or release something it was holding.
So you can trigger changes.
Exactly. It means these materials can be designed to respond to all sorts of triggers. Physical stimuli like changes in pH, or the presence of specific small molecules, maybe proteins, or even other nucleic acids, by building in specific DNA sequences that act as molecular triggers. So you could have like a tiny DNA box that only opens and releases its contents when it bumps into a specific cancer cell marker, for example.
Wow. That's incredible. Programmable, tunable, and dynamic. Okay, let's switch gears a bit. Let's talk electronics. You might just assume DNA, biological molecule. Must be an insulator, right? But what's the actual story there? Is it conductive?
Well, it's actually surprisingly complex, and honestly, it's still a topic of quite a bit of debate in the research community. Electron transport through DNA. It can show behaviors that range all the way from insulating to, well, somewhat conductive. It really depends heavily on things like the specific DNA sequence and the environment it's in.
So it's not straightforward.
Not at all. Think of it less like a copper wire, a superhighway for electrons, and maybe more like a series of stepping stones. There's a mechanism called charge hopping, where electrons jump short distances between the DNA bases. But sometimes imperfections in the DNA structure itself, what's called backbone disorder, can actually trap electrons. Localization.
Okay, so it's definitely not going to replace copper wires in our walls.
No, definitely not.
But here's the crucial part, and maybe this is the key insight for nanoelectronics. Its main role isn't really as an intrinsic conductor itself, but as a precise template or scaffold. What does that mean in practice?
Right. It means we use the DNA structure as a molecular blueprint to build conductive pathways, but out of other materials.
I see.
For example, scientists can take these DNA nanostructures and coat them, metallize them, with materials like gold or silver or copper. The DNA framework forces the metal atoms to deposit only along the DNA structure, forming incredibly precise conductive nanowires or other tiny electronic components, things that would be basically impossible to make using traditional top-down methods.
So the DNA draws the lines for the metal.
Exactly. Or you can use DNA to precisely position other conductive nanomaterials, like carbon nanotubes, maybe for building tiny field effect transistors. The DNA isn't the active electronic part, it's the perfect nanoscale assembler or mold.
That's a total game changer for building things from the bottom up, isn't it? Okay, let's connect this now to light. How does DNA origami play with light? What can it do in photonics?
Its role in photonics is equally exciting, really. DNA origami is used extensively now to create highly precise plasmonic and photonic devices.
Plasmonic. That involves metals interacting with light, right?
Yes, exactly. By using DNA origami to arrange metallic nanoparticles, usually tiny bits of gold or silver, with nanometer precision.
Which you can do because of the programmability.
Precisely. Because of the programmability, you can precisely control how these nanoparticles interact with light, tuning their plasmonic properties. This allows you to create things like ultra-sensitive hotspots for techniques like SIRS, that's surface-enhanced Raman spectroscopy.
Okay, and what's that good for?
It's incredibly useful for detecting very, very tiny amounts of specific molecules. Think diagnostics, environmental sensing. DNA origami also lets us build chiral structures, structures that interact differently with left and right-handed polarized light. This gives tunable circular dichroism, which has potential applications in things like optical switches or advanced sensors.
Wow.
And beyond metals, DNA origami is a fantastic platform for just arranging other light-active molecules like fluorophores, things that glow.
Okay.
You can position them with exact distances and orientations to study and control energy transfer between them, like A-bars T, first-order resonance, energy transfer. This is key for things like molecular scale photonic circuits or advanced biosensors that light up in response to something specific.
It really sounds like the ultimate toolkit for building almost anything at the nanoscale.
Yeah.
So let's bring it back to the real world. What does this all mean for us? What are the really exciting applications either happening now or just around the corner?
Yeah, the applications are really diverse and potentially very impactful. Think about nanoelectronic circuits first off. As we said, DNA is this amazing template for bottom-up assembly of electronic components, building connections, potentially leading to much smaller, denser circuits.
Okay, electronics, what else?
Then there are biosensing platforms. Because DNA is so programmable, you can design structures that bind very specifically to certain biological targets.
Oh, like diseases.
Exactly. You could detect pathogens, maybe specific disease biomarkers like early cancer markers, potentially much faster, more sensitively, and more precisely than current lab tests.
That sounds huge for medicine.
It is.
Yeah.
And speaking of medicine, drug delivery vehicles are another massive area. People are developing DNA origami structures as smart containers. They can encapsulate a drug, like a chemotherapy agent, and be designed to only open up and release that drug when they encounter a specific biological signal inside the body, like near a tumor cell.
So targeted delivery, fewer side effects?
That's the goal, yes. Minimize side effects, maximize effectiveness. And maybe the most futuristic sounding one is molecular computing.
Computing with DNA?
Yeah, harnessing DNA's natural information processing ability, the base pairing, strand displacement, to build actual logic gates and circuits out of DNA molecules.
What would you use that for?
Well, imagine tiny biocompatible computers that could operate inside our cells, maybe for complex diagnostics or triggering therapeutic responses automatically. It's still early days, but the potential is there.
Okay, this is amazing stuff. But it does raise a question. We already have these other 2D materials, right? Like graphene, MS2, molybdenum disulfide. How do these DNA-based materials really stack up against those sort of more established inorganic giants? What's DNA's unique edge, or where does it fall short?
That's a really critical comparison to make. Let's break it down. First, think about synthesis methods, how they're made. DNA relies on this amazing bottom-up self-assembly. You design the strands, mix them, and they basically build themselves based on the code you wrote.
Right, the programmability again.
Exactly. Inorganic materials like graphene often use top-down methods like exfoliation, literally peeling layers off graphite or bottom-up. But different methods like chemical vapor deposition, CVD, these can be complex, sometimes require harsh conditions or high temperatures.
Okay, so different ways of making them. What else?
Well, the biggest difference, arguably, is programmability. DNA has a clear, really unmatched advantage here. The base pairing rules give you this incredibly precise, programmable control over the shape, the size, the function, attaching things exactly where you want them.
Much harder with graphene.
Much harder, yeah. You have much more limited control over the exact atomic structure and functionalization with most inorganic counterparts. Then there's biocompatibility.
That seems like a big one for DNA.
Huge. DNA is inherently biological. It's part of us.
Yeah.
That makes it uniquely suited for applications inside living organisms, in vivo. Some inorganic materials, like graphene, still have question marks around potential toxicity.
Okay, but what about downsides for DNA?
Right. There are trade-offs. A major one is stability. DNA structures can be vulnerable to certain enzymes in biological environments called nucleases that are designed to break down DNA.
So they could get degraded.
They could, yeah. So researchers need to use various stabilization techniques, chemical modifications, or protective coatings. Inorganic 2D materials generally are much more chemically inert and structurally robust.
Makes sense. What about making lots of it? And cost?
That's another key point,
Right.
Scalability and cost. Synthesizing highly pure custom DNA strands, such as the long scaffold strands and hundreds of staples for origami, can still be quite expensive. Whereas for materials like graphene, large-scale production methods are more developed, potentially bringing down the cost for bulk applications. So DNA wins on precision and biocompatibility, but faces challenges in stability and cost scale compared to some inorganics.
Okay, that's a really clear comparison. So high precision, biocompatibility, those are DNA's superpowers. Stability and cost are maybe its kryptonite, at least right now.
That's a good way to put it, yeah.
So like any groundbreaking tech, there are hurdles. What are the main challenges, the big limitations that are kind of holding back the widespread use of 2D DNA materials today?
You've actually hit on some key ones already. That environmental stability is a big one. Dealing with those nucleuses, ensuring the structures survive long enough to do their job, especially for any medical applications, is crucial.
Okay, stability.
Then, as we just discussed, production cost. Making these complex DNA structures affordable, especially at scale, is a major economic hurdle.
Right, cost is always a factor.
Integration is another significant challenge. How do you actually connect these incredibly tiny nanoscale DNA devices or structures to the macroscopic world, to the wires, the readouts, the delivery systems we use? Bridging that scale gap is non-trivial.
Yeah, I could see that. Tiny structure, big world.
Exactly. And then there are other limitations too, like inherent error rates during the self-assembly process. It's not always 100% perfect. The complexity of trying to synthesize these things inside a living system in vivo is another huge challenge. And just figuring out efficient, large-scale manufacturing processes across multiple scales.
Lots to work on then. So, okay, pulling all this together, what does the future look like? Where do you see the really exciting breakthroughs happening next in this field?
I think we're going to see a huge push towards developing even more sophisticated dynamic and reconfigurable materials. Imagine molecular machines made of DNA. They can perform really complex sequences of tasks, changing their shape or function in response to multiple subtle cues in their environment. Much smarter systems.
Like tiny robots getting more complex.
Kind of, yeah. Another really big area is creating hybrid DNA inorganic systems. The idea here is to combine the best of both worlds. Use DNA for its amazing programmability and self-assembly to precisely structure inorganic components that might have superior electronic, optical, or catalytic properties.
So DNA builds the frame, something else does the heavy lifting.
In a sense, yes, leveraging the strengths of both material types. And of course, just continued advancements in those DNA-based molecular machines for all sorts of applications, diagnostics, therapeutics, maybe even nanoscale manufacturing itself. Pushing the boundaries of what we can build and control at that tiny scale.
Wow. Okay, we have taken quite a journey here. From the basic ideas, the foundations of DNA origami, how we can actually build things using our own genetic code, all the way through to these mind-bending applications in electronics, medicine, computing, and also facing the real-world challenges that researchers are tackling right now. It's genuinely amazing to think about our own genetic material being repurposed in this way.
It really is.
So let me leave you, our listener, with a final thought to maybe chew on. Given DNA's incredible, unparalleled programmability and its natural ability to self-assemble with such staggering precision, could it, maybe one day, form the actual literal blueprint for all future nanoscale manufacturing? Could we see biology and technology merge in ways we can barely even imagine right now?
Something to think about.
Definitely. Well, thank you for joining us on this deep dive. As always, keep asking questions, keep exploring, and most importantly, keep being curious.